Right, so today I will introduce several packages we developed for data integration and visualization of phylogenetic tree. Right, as we know, the Nui and Nexus are the two most commonly used tree file for storing the tree structure. And these formats are not human friendly, and there are many other formats that are not compatible with each other, especially for some software output, and they cannot be recognized by other software and even by the software itself. And there are some heterogeneous associated data, including phenotypic data, experimental data, clinical data, and analysis finding that we want to link to the tree structure and analyze and interpret this data in the evolutionary context. And the third problem is we want to present the data on a tree. So this is what we call annotating a tree with associated data. And there are some software, including figure tree, ITOL, EvolveView, etc. But these tools are all restricted to predefined annotating utility, and they focus on specific analysis and domain-specific data. And they contain non-overlapping features and are not compatible with each other, and they are hard to customize. So to solve this problem and issue, we developed several R and bioconductor packages, Quen and bioconductor packages to solve this problem, including treeio for importing and exporting tree with associated data, tidytree, which provides a tidy interface, just similar to like the prior, to manipulate tree data, and GT3 for visualizing and annotating phylogenetic tree with heterogeneous data. And the GT3 etcher, they're able to present data on a circular layout. All these packages, except tidytree, are on bioconductor, and tidytree is available on Quen. And this figure shows the main features providing in the treeio packages. We provide utilities to pass many tree file formats like the NXJ place, Philip, etc., including Nexus and Nui. And we are able to pass software output that contains tree and their analysis results, including many software that are widely used in evolutionary biology. And this software output are hard to pass, and most of the software can recognize this software output. And all the data after reading in R will store in an object called tree data, which contains the tree structure and their associated data. And we provide some function to manipulate the data. And the treeio packages are able to output the tree data object to a single object, including these compatible nexus file, J3 and Nui and Nexus. This allows us to convert different software output and allow us to combine external data with the tree structure. And here is an example. We use a bees output and using the ReadBase function to pass the file. After passing the file, all the statistical inference will be stored in the tree object, and then we can analyze the data in R and also present the data using GT3. And suppose we have a data is from external sources, then we can link the data to the tree structure, and the data will also incorporated in the tree structure, in the tree object. And treeio also provide a merge tree function to combine two tree objects. So here we use bees to analyze a tree, and using CoML to analyze the same tree. So we have two results. Then we use read.bees and read.coml to pass this tree file. And then we merge them into one single object. Then all the statistical inference from bees and from CoML are merged together and stored in a single tree object, including the divergent time inferred by bees and the selection pressure inferred by CoML, and also the ancestral sequence inferred by CoML are all available in a single tree object. This allows us to combine analysis results from different software using different models, and allow us to compare different model or compare the consistency of different software analyzing using identical model. And as we know, the tree structure is hard to manipulate, and we provide a tidytree package that can convert a tree object to a tidy data frame. So this is very familiar as by most users. We can even export this data frame to a CSV file and process your tree data in itself. Now this removes the barrier for ordinary user to process the tree structure and their associated data. And after processing the data, we can convert the tree object back to we can convert the data frame back to a tree object, either the bare tree or the tree with their associated data in tree data object. So here is an example, which is a question posted in the GT3 Google group, and we want to insert a knot between two knots, and we want to display some uncertainty. This is difficult to do, but with the tidytree package, it's quite easy, as we can convert the tree to a data frame, and then we can add another column to differentiate the newly introduced knot and other knots, and to add the information of the uncertainty, and then we can visualize this tree using GT3. The treeio package allow us to export tree with data to a single tree file. So this allows us to convert software output and to combine tree with external data, and also merging tree data from different sources. And the screenshot here shows two examples. The tree inferred by the CoML and by Phylogenetic, they cannot be recognized, cannot be opened in figure 3. But after we reading the data in R and then export it to a Bees Nexus file, it can be opened by figure 3, and we can use the evolutionary inference performed by these two software to color the tree branch. So it allows us to convert software output and make the data more widely available for other downstream software. Okay, now we move on to the GT3 package. The GT3 supports tree data passed by the treeio and also other tree-like structure, and it allows us to integrate variety type and sources of data. It supports the grammar of graphic for high-level customization, and it allows us to manipulate and exporting the tree visually. here are all the tree layout that's supported by GT3, and here we also suppose visualizing tree in 2 dimensions. That means we use another variable to scale the y-axis. And in this example, we use the predicted and linear causation side of the HA influenza virus, and we can, and this figure shows very clear pattern that the number of the glycosylation site of the HA protein are quite different from the virus infected swine and human. GT3 is fully compatible with ggport2, and it supports the grammar of graphic. And here is the example of how we use GT3. After we pass a file and it's stored in the tree object, we use the GT3 command to show the tree structure. And then we use the operator plus to add annotation layer. And in this example, we add a layer of the tip label and another layer in the middle of the branch to display the species name, and another layer to display the duplication event, and another layer to show the bootstrap value. This is what we do in GT3. We only provide Geo Layers that serve as the building block, and users are free to combine them to create a complex tree figure in the way that they want. And here is a table listing all the Geo layer provided in GT3, and we are not restricted to use these Geo Layers. Other Geo Layers defined in ggport2 and other ggport2 extension are, most of them are compatible with GT3, and we can use them directly in GT3. And just like here, the Geo Layer, Geo Text are all provided by the ggport2, and we can use them directly in GT3. GT3 also supports overlay image file on a tree, and we support querying scientific name from the Phylogenetic database, and then download the figures automatically and display in on the tree. And we are able to use associated data to, for example, to color the image or scale the size of the images. And GT3 supports annotating tree with subplots. So here, as an example, we use pyplot to display ancestral states for categorical variable. If the ancestral state are in numerical value, we suppose using continuous state transition in S. So in this figure, we can see very clear how the value was transit from the ancestral state to their offsprings. And GT3 supports plotting many trees at once as a density tree or in different panels. And it also supports visualizing 2 trees face by face for comparison. GT3 is not only for phylogenetic tree. It actually supports almost all tree-like objects defined in the R community, including tree graph and hierarchical clustering. So almost all tree-like structure can be visualized using GT3, not only for phylogenetic tree. And the output of GT3 is a GT3 object. And the object maintains the integrity of annotation data and visual style derivatives. And in this example, we use a very simple tree and using GT3 to display the figure here. So the output is stored in the P object, and this P object contains all the information. So we can convert it back to a tree object. So here we use as the tree data to convert a graphic object to a tree object, and this tree object contains all the associated data and also the tree structure. We can also export the tree structure to a GUI text. And the graphic object also contains all the visual style derivatives. So here we have another tree object, and we provide an operator, this operator to in GT3, and we can use the visual style derivative to visualize this new tree object. So here is what we get. This is very similar to format painter in Microsoft Word. So we use we can apply a visual style from a tree object to from a graphic object to another tree object. So here is the last part. We move on to the tree and to plotting data on the tree. For this problem, we can they're actually divided into two categories. First of all, we want to mapping the data to the tree structure to display the data on the tree directly, or use the data as a visual characteristic of the tree branch and knots. For example, like the line size, color, and knot shape, etc. And another problem is we want to align a graph to the tree based on the tree structure. So we may be able to visualize our data as a schema, as a dot plot, a bar plot, a line plot, etc. But if we want to align the graph with the tree, the plot needs to reorder based on the tree structure so that we can interpret the data presented in the right-hand side panel using the tree structure and interpret them in the phylogenetic context. For the first question, we define an operator, the attach operator, to attach external data to the graphic object. And after attaching the data will be served as a global variable and visible to all the Geo Layers. So here are the example we use this operator to attach information of the tip and external knots, and then we are able to using this data to annotate the tree. And for the second part, we provide a basic plot function, and this function will eternally reorder the input data based on the tree structure, and then using the input Geo layer to visualize this data in a different panel. If we progressively call the basic plot to visualizing different data, it will display the data in different panels. And here are some examples of using this basic plot. So we don't need to care about the order of your data. The basic plot will do it for you. It will automatically reorder the data based on the tree structure so that we can interpret the data in the evolutionary context. The basic plot function only works for rectangular layout, so we develop another package called GT3 etcher to help users to present data on circular layout. And here are two examples. This package is developed by PhD student Shuangbing, and he will introduce another package, the microbiota process, that also developed by him, and he will introduce the package, the uses of the package for analyzing microbiome data. And here are the publication in all these packages. And here goes the summary. We also draft a book that introduce all the detail of these packages. And the book is online, and freely available online. Thanks everyone for your attention. Questions are welcome.
Thank you so much, Dr. Yu. We have one question from Julie. So when merging trees, do you need to scale the input trees first?
Okay. What's the scale means? I can't get the idea.
Julie, you can unmute yourself.
Also a great talk and great package. And so for scale, I meant, you know, if you draw the tree, think about the length, the distance, like a phylogenetic tree, the distance between different species, right? So when you merge two different trees, are they scale similarly? If not, are you going to, that's my question. Second question, I'm just going to talk about what's the underlying algorithm to automatically place nodes on the drawing panel relative to each other, and what algorithm you use, I guess. So I see a very different, clustering, very nice, aesthetically beautiful. It's automatically drawn, right? You don't have to do much. How you do that within this underlying algorithm? Thank you.
Oh, okay, I get the idea now. First, for the first question, for scale the tree, after we merge the tree object, actually we in GT3 we are able to use different variable to scale the tree. So after merging the tree, all the associated data and branch length in different software are all stored in the tree object, and we can use this variable to scale the tree. I mean the branch length, right? So actually we can okay. There is a figure presented in the ME paper. Okay, let me see. There is an example presented in this figure that it helps us to exploring the tree data. We are able to specify which variable to scale the tree. So for the second question, do you mean here using how we reorder the data?
So some of the very fancy ones that you have a cluster, some cluster on the left, some cluster on the right, they are very aesthetically appealing, not just like aligned. So is that automatically generated either somewhere? Go back, I think. Let's go back. Go top. Go top again. Let me see. Keep going. Keep going. Keep going. Yeah, something like this. It's almost like a trajectory, right? How you go, like, is that, and also another one before that too, I think even more simpler one. Maybe go up again for a simpler one. Go ahead, go, continue. Yes. Oh, sorry. Yeah, this type of thing, you have left side, right side, and nicely clustered, and then the branch drawing away that not all of them on the left or all of them on the right. It's like a spatial arrangement. Is that automatically placed? You have an algorithm to do that?
You mean clustering here to label the clay?
Yeah, right.
No, we did not have. We can do it automatically. We need to specify which clay you want to highlight.
Oh, and how about on the left side? Like you have, you know, those two on the right, one bigger one on the left, and then one bottom one. Do you have to specify this branch or line here, not the, you know, on the other side, all together clumped?
Which one?
For example, the left side, right? You have a big highlighted peach color, and then you have purple and green, like purple green on the right side, right? And you can easily think about it, the blue can go overlap on the purple, which will not look as beautiful, right? If blue, you can move the branch around to be placed on near purple or in green, which is not going to be good because all clustered together. So I'm wondering if you did that automatically or you actually specify a parameter to make it place on the right corner there.
We need to specify parameters.
Okay, you did, okay. So I just wondering if you have that, okay. No, it's fine, okay. I thought you have some kind of algorithm to place that, you know, beautifully. Okay, thank you. That's all my questions.